Okay, so now we are going to look at the examples of the secondary structure of protein. So the secondary structure of proteins, we have alpha helix and beta-pleated sheets. So now we are going to start with the alpha helix. So in the alpha helix, it has a regular helical conformation that is rod in shape. So it means that they have rod shape. And in alpha helix, the carbonyl oxygen is hydrogen bonded to hydrogen on the amino group of the fourth amino acid. So it means that in the beta-pleated sheets, there is a hydrogen bonding between the carbonyl oxygen. Remember, in amino acid, we have carboxylic group, and this carboxylic group is made of one of the carbonyl compound. And this carbonyl compound is carboxylic acid. So this is the functional group of carboxylic acid. So it means that when in the case of alpha helix, this carbonyl oxygen, it usually forms a hydrogen bonding with the amino group of the fourth amino acids, and therefore 3.6 amino acid per ton of the helix. So therefore, in 1 ton of the alpha helix, it means that there is 3.6 amino acids, and it covers a distance of about 0.54 nanometer, which is exactly, so therefore in this indicate that each amino acid residue represents 0.15 nanometer advance along alpha helix axis. Then the side chain is positioned is outside of the helical structure. So this is actually a typical example or a typical demonstration of um, alpha helix of an amino acid, and the distance of this amino acid is uh, is uh, is 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 1.5 Armstrong. And then per ton of the alpha helix is about 3.6 amino acids. Okay, so now this is also another example. It's actually the same diagram. This is how the alpha helix in the secondary structure of proteins look like. Then the next thing is beta-pleated sheet. So in the beta-pleated sheet, the hydrogen bonds form between the peptide bonds. So the hydrogen bond is formed between the peptide bond, either different polypeptide chains. So a different polypeptide chains in different polypeptide chains, the hydrogen bond may be formed in the case of beta-pleated sheet, or in different section of the same polypeptides. So it means that maybe when you have different polypeptide like this, you may have actually hydrogen bonding exists between these polypeptides. Like for example, let's say this is one polypeptide and this is another polypeptide. So therefore, the hydrogen bond in beta-pleated sheet can be formed between different polypeptide chains, or it can also form the hydrogen bond at different sections, different parts of the same polypeptide chain. And then it is a planar, that is the planetary of the peptide bonds, direct the polypeptides, and deplete it with the one or with the sides chain amino acid protruding above and below the sheets. Adjacent polypeptide chain in beta-pleated sheet can be either parallel, antiparallel, depending on the directions. So in the beta-pleated sheet, the polypeptide chains can either be in a parallel direction or antiparallel direction, and it is fully extended with a distance of 0.35 nanometer from alpha carbon atom to the next, and the sheet slightly curved And if several polypeptides are involved, then the sheet can form what we call beta barrel. So several polypeptide chains, if it moves in forming the beta-pleated sheet, then it may actually change a form to what we call a beta barrel. The multiple beta-pleated sheet give strength and rigidity in structural proteins. So it means that this beta-pleated sheet, if it founds in a particular proteins, it gives the strength and the rigidity of a particular structural proteins like keratin. Then is examples of the structural proteins. We have like fibroin, which consists of antiparallel beta-related sheet stacks. Okay, so this is actually how it looks. So for the parallel, this is for the parallel. This is for the parallel. So you can now clearly see it's actually the secondary structure of proteins is made of some components. It has hydrophobic component and hydrophilic component. There is some part of it will interact with water while some will not. Then so parallel beta later sheets means that have the same direction of N and C terminal end of peptide. That is two polypeptide chains run in the same direction. So if you have two polypeptide chain run in the same direction, then we call that as parallel. Like for example, this is polypeptide, so it moves in this direction. And this one is also another one, moves in the same direction. That is why it's called parallel. This one also is the same direction, the same direction. This one and this one are in the same direction. So this is what you call parallel. In the case of antiparallel beta-plated sheet, it means we are talking about opposite direction of N and C terminal end peptides. There is two polypeptide chains that run in opposite direction. That is anti-parallel nature of beta-plated sheets. This is how the beta-plated sheets, both the parallel and the parallel structure. So this is how it looks like. The comparisons of alpha helix and beta-plated sheet secondary structure of proteins. So coiled rod-like, it looks like coiled rod-like, and this one is fully extended sheet-like. Then the hydrogen bonds are parallel to polypeptide backbone in the case of alpha helix, and here the hydrogen bond are perpendicular to polypeptide backbone. The axial distance between amino acid is 1.5 Armstrong, while the axial distance between amino acid is 3.5 Armstrong. So one constitutes polypeptide chain. So, and then now we are also going to look at the tertiary structure of protein. So in the case of tertiary structure of proteins, it's just a spatial arrangement of amino acid far apart in a linear sequence as well as a residue adjacent. So in a tertiary structure, we are just talking about the three-dimensional conformation of a polypeptide. So this indicates that it is in a 3D space. How secondary structural features accessible, then form a domain and how these domains relatively spatially to one another. So you see, in the case of the tertiary structure, we're just talking about the three-dimensional structure of proteins. And this three-dimensional structure of the proteins is actually based on the number of domains as well as the spatial arrangement of the atom. So a domain is a section of protein structures that is sufficient to perform a particular chemical or physical task. Like for example, binding of a substance or binding of a substrate or other ligand. So a domain can have the ability to bind to a substrate or ligand. Example of the 3D or tertiary structure of proteins, we have myoglobin. So myoglobin is a globular protein where the peptide change folds spontaneously so that majority of its polar charge side change amino acids are on the surface and majority of the hydrophobic chain are buried in or are buried in the interior. So this is in the case of myoglobin. Remember, I think in some previous videos we talk about the different types of proteins based on their solubility. So we have globular protein, and then we also have what do you call fibrous proteins. So in the case of globular proteins, what usually happen is those amino acids that are hydrophilic in nature will be exposed to the surface of the proteins, while those are non-polar amino acid, which is hydrophobic amino acid, will be buried in the interior so that at least it will not have actually access to the water molecule. So once folded, the three-dimensionally active, that is native conformation of a protein, is maintained by the non-covalent forces. And these non-covalent forces, you should know them, ladies and gentlemen. We have hydrophobic interactions, we have hydrogen bonding, we have electrostatic force, as well as a covalent disulfide bond. So these are the four processes that actually make the tertiary structure proteins to be fully folded and maintain its three-dimensional structure. These are the examples here. We have beta-pleated sheets, then we have alpha. So look at the structure. So in the case of alpha, it looks like it is spiral. It is spiral. Then for the beta-pleated sheet, it looks like a mat. All these bonds, different rearrangement from the primary structure of proteins, is actually based on the non-covalent forces, which is hydrogen bonding, electrostatic force of attraction, and the rest. Okay? Then for the quaternary structure, we are talking about one polypeptide, greater than one polypeptide. It must be greater than one polypeptide chain, such as hemoglobin. So hemoglobin is also at its quaternary as well as tetrahedral structure, and the spatial arrangement of polypeptide subunits and the nature of the interaction between them. So these interactions may be covalent linked or non-covalent interaction. So you can have actually different spatial arrangement of these polypeptide subunits and the nature of the interaction between them. So these interactions that are usually found in the quaternary structure proteins is the covalent links and non-covalent links. And as I also explained what the concept of those other non-covalent pulses. So this is actually how the quaternary structure of proteins look like. This is also another one is for hemoglobin. So hemoglobin has four polypeptide units. So it is tetramer, yes, and it's associated by non-covalent bonds, which is alpha to alpha chain and to beta chain, and it possesses quaternary structure, and each chain has one heme group and one ion, two ion. Okay. So this is actually how the amino acid look like. It's like our bricks. This is the primary structure, secondary structure, tertiary structure, and then the quaternary structure. So there are four orders of this protein structure. There's a bricks, and this is the bricks. That's the raw amino acid. And then the wall. The wall is the primary structure. And then bent. twisted in that walls is the secondary structure. And then the tertiary structure, that is safe contained room. It's a self-contained room because all the bonding that happened is actually as a result of some basic interaction between uh yeah, okay. So I think for this we just need to understand how this structure of proteins look like. So I'm going to stop here and then we are going to watch another video